Syncell Microscoop technology enables the discovery of spatial protein constituents, making de novo subcellular spatial proteomics feasible for the first time. Microscoop is the only tool for unbiased spatial proteomics, requiring no predetermined protein antibodies. It reveals low-abundant proteins, discovers ones that previously were not known, and enables studies focused on drug-targeted protein locations rather than genes. Regions of interest are areas where researchers want to identify all proteins present, ranging from subcellular structures approximately 250 nanometers to entire cells. Cells are seated in an imaging chamber and fixed to preserve their structure. These ROIs are defined using 1 to 3 fluorescent markers or other visual landmarks visible under a microscope. If you can clearly define your region of interest under a microscope using immunofluorescent imaging, Microscoop can achieve targeted region labeling. Microscoop has been successfully used with formalin-fixed paraffin-embedded, fresh frozen tissue samples, and fixed cultured and primary cell samples. Our Synlight-rich kit, containing photochemical probes for the photolabeling reaction, is first added to the sample. After the sample is loaded onto the stage, Microscoop takes an image of the sample at one field of view at a time. The image is analyzed in real time by Microscope's software Autoscoop, which executes traditional image processing or AI deep learning to segment and define the user's region of interest. A femtosecond light source precisely illuminates the segmented region of interest, one point at a time. The Synlight kit contains photoactivatable probes that biotinylate the sample when light is shown at high resolution. A focused 2-photon laser triggers the biotin-based probe within the region of interest, binding biotin covalently to proteins colocalized with the target. This patterned photolabeling is repeated for hundreds to thousands of FOVs automatically to ensure that enough proteins are biotinylated for later proteomics analysis using mass spectrometry. Photolabeled cells or tissues are scraped from the slide or chamber. Samples from multiple slides or chambers can be pulled together to increase the total protein amount. The scraped sample undergoes lysis with the Synpool kit, followed by protein enrichment through streptavidin biotin pull-down. Streptavidin beads selectively bind the biotinylated proteins, which are then digested into peptides for detailed proteomic analysis. The collected peptides can be sent to a mass spectrometer to perform LC-MS analysis. The protein lysate is also compatible with other proteomic readout techniques. Proteomes of both the photolabeled and unlabeled, our control, samples are obtained. By comparing the control and photolabeled proteomes, a location-specific proteome at the region of interest is obtained in high sensitivity, high specificity, and high spatial precision.